All right, welcome to Life in Biology. I'm Dr. Joel Graff. This is some slide. We're going to go over some slides today that were drawn in my lecture on Wednesday, February 2nd, 2022. So we had just been talking about the genetic code. We also drew pictures of ribosomes sitting on an mRNA in the act of translating. And one of the key parts of the figure that I left out at this point was the tRNAs. It would have been very messy to try to draw tRNAs in each of those three sites on the large subunit of the ribosome. And so what I'm doing now is I'm going to just take a second to talk about the tRNA and its structure. tRNA stands for transfer RNA, and what's it transferring? It's going to transfer amino acids. So as you're making a longer and longer amino acid chain to make a protein, each tRNA that is involved brings one amino acid in. The way that tRNAs work is that they have three nucleotides in a particular part of their structure that will read the mRNA, and then at the other end they've got one amino acid, and so it's the tool for reading RNA and attaching the correct amino acid. Now, we talked about the genetic code and how there was 64 possible codons. A, C, G, and U are possibilities for the first letter, second letter, and third letter. So that's 4 to the third power, and that equals 64. Now, 3 of the codons were what designated stop codons, and they don't actually code for a particular amino acid. Instead, it's not even a tRNA that will show up when you have a stop codon there. It's something called a release factor, and we'll go into that in more detail later in the course. But anyway, so there's 64 codons, 3 stop codons, which means that there are 61 unique tRNAs being made in the cell, and each of those tRNAs will recognize the 61 codons, and so 61 different tRNAs. So here I've drawn a structure. It's a single-stranded RNA. I don't remember which strand of the RNA is the 5' end and 3' end, so I left it off, but one of these is a 5' end. One of them is a 3' end. Anyway, there will be an amino acid stuck to one of the ends of the RNA, and this particular amino acid is going to be specific to this version of the particular tRNA. So there's going to be machinery in the cell that will match up amino acids and charge tRNAs, that is, put an amino acid onto a tRNA if there's a tRNA that doesn't have an amino acid. So that charging of the amino acid brings in some of the specificity in the system. Now, again, because we're switching from the drawing on a smartboard to then saving it as a PowerPoint, this is kind of hard to read. These 3 letters are U, A, and C. So in these 3 particular letters, these 3 nucleotides are going to be called the anticodon. And the anticodon will match up with its codon. And so opposite of a U would be an A, opposite of an A would be a U, and opposite of a C would be a G. And so here I have UAC, and I have them connected to AUG. So this particular anticodon, UAC, will then match up with RNA sequence that is AUG. And so that is what is meant by the anticodon. So for the 61 different tRNAs you have 61 different possible anticodons here to match up with the 61 codons that we have after we subtract the 3 stop codons from our total. Okay? So anyway, you have this single-strand RNA, and it has this funky structure where it will form some stems, and you also have some parts that form the loops, and I had gone over RNA folding in the previous lecture. But anyway, it kind of looks like a clover leaf, but actually that's how you would draw it on a 2-dimensional screen like I did for class. In reality, these actually fold up into kind of a tube shape these, if you want to consider these arms, they're kind of tucked in like a figure skater doing a spin. And so then it has a general just bent shape like this. So we can just, and on the Khan Academy, they will draw the tRNAs as kind of a dumbed down version where you've got anticodon here, amino acid here, but that's just this structure but kind of folded up. Okay, so what good is the tRNA? Remember, on the ribosome, we have the A, the P, and the E sites. The tRNAs will, with the amino acids, will come into play when a codon becomes exposed in the A site that matches the anticodon. So whatever tRNA should match the codon, has the anticodon to match the codon, will jump into that site, and then the amino acid that it's holding on to will be connected, covalent bond, peptide bond, to the growing polypeptide chain that was currently in the P site. If you can't remember A, P, and E, that's okay. I have a link here to a video. The video is called From DNA to Protein. It's a nice little animation. The YouTube channel is called Your Genome. And if you have access to my slides, you can use this hyperlink. If you don't have access to this slide, between this title and the channel name, you should be able to find it. But the reason I show this is because it shows a lot of the things that we've talked about so far. It starts with DNA, and it talks about the transcription bubble, and we drew pictures of the transcription bubble before. It also talks about how the ribosome moves down an mRNA 3 nucleotides at a time, and how each time it does that, it brings in a new tRNA. And so it's just very difficult to just draw static pictures of this whole process. So I think it's very much worth your time to take a second to go look at animations. And this is just one of many animations on YouTube, but it was only 2.5 minutes, so that's why I picked it. It was nice and short and got to the point fairly quickly. Okay, so then when we make a protein, it's going to fold up into some complex three-dimensional shape. But we also know that we could describe the protein as a sequence of amino acids from the N-terminus to the C-terminus. And so when we talk about protein structure, we break it down into 4 levels. There's a primary protein structure, which the shorthand for primary is a 1 with a degree sign. And this is going to be everything from the amino terminus to the carboxyl terminus, the N-terminus and C-terminus, if you want to use shorthand, the amino acid sequence. So this is likely a methionine, and then this is one of 20 different amino acids. Here's another of 20 different amino acids, and so on and so forth. I drew little dots in here to indicate where some peptide bonds form. Here is a generic amino acid. I'm focusing on the alpha carbon in the center of the amino acid. We got the amino group to the left, the carboxyl group to the right, a hydrogen up, and an R group at the bottom. And when you have 20 different amino acids, it's the R group that's going to be different among them. So anyway, the carboxyl group here, when the next amino group comes in, is going to you're going to connect the amino group of the next amino acid to the carboxyl group of the current amino acid, and so they get connected together. So there's only one end that has an amino group sticking out by itself, and the other end has a carboxyl group sticking out by itself, and so that's why we talk about it in N-terminus to C-terminus. It's kind of the same as 5' and 3' for DNA and RNA. When you read the mRNA in a 5' to 3' direction, you make the amino acid chain from the amino terminus to the carboxyl terminus. Okay, so that the actual just sequence of amino acids from the first amino acid to the last amino acid is the primary structure. In the secondary structure, which you can use shorthand a 2 with a degree sign, there's 2 main types of secondary structure. One is called an alpha helix, and so in that case, the amino acids start spiraling into a helix shape. In the other type, you've got several strands of amino acids that will zigzag back and forth on themselves. Each of those strands is called a beta strand, and they form together interactions from this strand to this strand and interactions from this strand to this strand into a what's known as a beta pleated sheet. It's pleated because it has a zigzag pattern in the way that it's arranged. Okay, so anyway, when you talk about secondary structure for the beta part, you can think about the strands. You can understand that they can go together to form sheets, and then once you get even further, you can have beta sandwich. So if you can have two beta sheets stacking on top of each other, it's a beta sandwich. However, I should point out that the beta sandwich would be then categorized in this next part, because in secondary structure, it's neighboring or very close by amino acids, backbone molecules interacting with each other to form that alpha helice or these beta sheets. As you have a very large protein, it can then fold up into complex shapes, and this three-dimensional folding versus these more or less linear looking structures there, the three-dimensional structure is the tertiary structure. And when a protein folds over, you can have amino acids from one end of the protein interacting with amino acids of the other end at these contact points. And in that way, it's going to typically be the R chains of the amino acids that are interacting with each other. So the R chains are, we learned that the amino acids, we could group them into four basic groups. We could have the nonpolar, the polar, the positive charge, and the negative charge. And so those polar molecules, those nonpolar molecules, and those charged molecules can all interact in different ways to form different kinds of bonds within here. You can even get some covalent bonds formed, but that's a special case that we might talk about later. Then it seems like we've covered everything. We're now in a three-dimensional structure, but the fourth level of folding, and this isn't true for every single protein, this is true for a subset of proteins, they go on to have quaternary structure. And in a quaternary structure, you can see that you've got three-dimensional molecule drawn here in this color, another one in this color, another one in a third, and another one in a fourth color. So this is four different polypeptide chains that are then interacting to form one protein. So a protein, we originally said, was just lots of amino acids stuck together. It's a little bit misleading to describe it like that, because sometimes proteins can be made of a single chain of amino acids stuck together, or if the protein has quaternary structure, there are a set of polypeptides that interact with each other. Each of the polypeptide is considered a subunit, and so then you have subunits of proteins. And so this is multiple amino acid chains stuck together to form the final protein structure. So it's a little bit confusing, but it's just to point out that not all proteins consist of one amino acid chain. Some proteins consist of subunits, where each subunit is a length of amino acids. Okay, so then we talk a lot about protein structure and folding because these turn into important molecules in the cell. And if you look at the central dogma, DNA encodes information that can be read and made into RNA. The RNA can be read and made into protein. So that's our central dogma, of course. But why are proteins the end of the line? What makes them special? Well, it's because they do a lot of different functions in the cell. So we can have enzymes in a cell. We've talked about enzymes previously, and we'll come back to them again when we get to metabolism. But enzymes are catalysts. They speed up chemical reactions. Hormones could be proteins that are used to communicate throughout the body. A third function for proteins would be transport. Maybe it's transport across a membrane or within a cell. I'm going to come back to that because I have motor proteins as a separate point. But anyway, getting molecules moved around in the body can be aided with proteins. Structure, there's certain proteins that'll form the cell cytoskeleton. Another example of structure from proteins is the capsid of viruses. So that's the protein coat that you find on some viruses. And finally, or not finally, number 5, we got 7 to go, or 7 total. Number 5 is defense. So there's all these different proteins that could be floating around either within your cells or outside of the cells. I've listed ones that are outside of the cell that can specifically stick to dangerous things in the body, and they start an immune response. I mentioned antibodies. Antibodies are an example of a protein where there is quaternary structure because it's made of 4 subunits, and or more depending on the class. Then I've mentioned complement. These are very common proteins you'll find in the blood, and they're part of your first line of defense if you get an infection. It can be either in the blood, or these proteins can seep out of the blood and into the tissue. And then there's also other proteins that might coat, say, a bacteria, and this makes them tasty to macrophages. That's called opsonization. We're going to come back to macrophages, which are big eaters. They eat things through a process called phagocytosis, and we'll be covering that in a couple days. Motor proteins, these are really cool. When a muscle contracts, or when you have a vacuole that needs to be drug around the cell somewhere, these motor proteins are the little engines behind the scene, and they oftentimes can walk along other motor protein, or they oftentimes walk along the cytoskeleton. Sorry, I found the word I'm looking for, cytoskeleton. So these motor proteins can walk along cytoskeletons, and that's how you can contract a muscle or move a gigantic vacuole around compared to the motor protein. But we'll watch an animation later on this semester where you can watch a motor protein pulling a vacuole along a microtubule, which is a type of cytoskeleton, and it's just amazing to watch this little protein basically take step after step as it's moving through the cell. And then finally, protein can be energy storage. And in that, the example I gave in class was when you have, say, a chicken egg that's been fertilized, the embryo will start growing, and the egg white, the albumin, is a source of energy for that growing chick. So that's an example of protein being used as energy storage. And there's probably more types of things that we can list about protein functions, but those are at least 7 big categories of things that proteins do in the cell, and why they're some of the most complex small molecules in the universe. Okay, and then for the next slide, we just had time to dig into our next topic, which is diffusion. In diffusion, the basic idea is that if you've got an area that's filled with fluid, and the fluid could be either liquid fluid or air fluid, basically anything but a solid, diffusion can happen. And what happens is that you will, for whatever reason, have some molecules that are all piled together, and then they are bumping around. The water molecules or air molecules, depending on if you're talking about liquid or gas, are bumping around as well. And so these molecules here will get jiggled around by the water, they'll bump into each other, and if you do lots of bumping like that, there's really nothing holding them back from getting spread out to the rest of the area. So if you give it enough time, they'll bump enough that they're this far apart, and then if you give it a really long amount of time, they'll even out so that there's no areas where there's a lot higher concentration than other areas. Okay, so diffusion. This is when molecules spread out. They move from areas of high concentration to areas of low concentration, and that's what I was drawing here. The energy needed to do this can be just the heat that's in the air. So as long as you're not at absolute zero, molecules are going to be jiggling around, and so that also means the hotter you are, the water or air, or the liquid or air is, the faster diffusion can happen because things are bouncing around. It's a form of kinetic energy, and these things are bouncing around and hitting each other faster. So diffusion will occur faster in warm fluids than in cold fluids. And then the last point, I really should have drawn this on a separate slide, but you have areas of high concentration to areas of low concentration. And so as you move from high concentration to low concentration, it's sometimes called the concentration gradient. So if you want molecules to move from high concentrations to low concentrations and even themselves out, as is shown here, you don't really need to add in any extra energy. It's just the heat is enough to do it. Whereas if you have molecules spread out in a low concentration and you want them to get packed together into high concentration, the only realistic way to do this type of thing is to add a bunch of energy to the system and basically corral the molecules and get them to be in a higher concentration. And we're moving on the next slide in the talk that we're given today in class. It's going to be about membranes and semi-permeable membranes, and we're going to be talking about how you can take molecules that can't go through the membrane and you take it from the inside of the cell, for example, where there might be a low concentration of, let's say, calcium, and then you use ATP to push a calcium out onto the surface of the cell. So there can be as much as 10,000 times more calcium outside of a cell than inside of a cell, and it takes a bunch of energy to get that gradient built up. I think that's the last slide. So yep, kind of a short talk. It took a long time to draw all of those things, and we watched the video as well. So anyway, this has been Life in Biology. I'm Joel Graff, and feel free to like and subscribe or not.